right, thank you and good morning. And you did hear a lot about Tennessee there, and I'm a huge fan. So if you follow football, you know what an excited person I am right now until at least after Saturday when we play Georgia. But you are very lucky. I do have an orange suit, but I did not wear it today because I was scared it would blind you all. But anyway, we'll get started here. So yeah, I'm going to talk about pharmacogenomics. Now I'm CPIC. I live CPIC, so I will focus most of this talk on that. But we have been having conversations with the FDA about ways to really advance the field, and I'll talk to you a little bit about some of that ongoing work as well. So I'm going to start with the patient case because I think this is good for this audience, especially to see before CPIC, what resources did we have? How did a physician or a pharmacist, how did they translate any type of pharmacogenomic knowledge into a prescribing recommendation? So I'm going to start by reading the case. So, and I've got an error in here, so I apologize for that. But AG is a 9-year-old female, or actually male, and is being treated for ALL and is immunocompromised. So he presents today with fever. The medical team suspects a fungal infection and would like to start voriconazole. So you note that the patient has a CYP2C19 genotype in the chart, and it's CYP2C19*17*17. So as the pharmacist on the team, what would you recommend to the patient regarding the use of voriconazole? So yes, I used this case at a pharmacy meeting, but I still thought it applied here. So this pharmacist goes to the drug label, right? And I'll be honest with you, I've never gone and pulled a drug label, okay? I usually go to LexiComp or Micromedics or one of the drug resources, but in this case, this pharmacist did. So she searched the label. What is in the FDA label about voriconazole and CYP2C19? And so this is a great resource. If you don't use Pharm.D, you should. It's a really great resource. You can go to that website, you can put in any gene, any drug, and you can find a lot of information. And one of the things they do offer are these annotations of the label. So, and it's great because you can compare the FDA label to the European label to the Japanese label. So it's really a great resource. So I just screenshot it. This is a screenshot from that. But she noticed that in the metabolism section, it does say there's some in vitro studies that showed voriconazole is metabolized by CYP2C19, CYP2C9, and 3A4. And there are also in vivo studies that indicated that CYP2C19 is significantly involved in the metabolism of voriconazole, and this enzyme exhibits genetic polymorphisms. Well, thank you. Yes, we now know this. But then it goes further, and there's a little bit more information in it. Then here you can see at the bottom here, it talks a little bit about populations, but then goes in and says that studies conducted in Caucasian and Japanese healthy subjects have shown that poor metabolizers have, on average, 4-fold higher concentrations exposure than their homozygous extensive metabolizers, and then subjects who are heterozygous have 2-fold. Okay, so the pharmacist goes, I don't know what that means. My patient's CYP2C19*17*17, it's not mentioned here, but I'm going to keep digging. What else is out there? So she finds this list of the FDA-approved labeling, that 120 drugs of pharmacogenomic information. She searches this list. Voriconazole is also not on this list. But in February of 2020, before the whole world exploded, I think we were making good strides at that point, and then of course the FDA definitely got busy with COVID. But they put out 3 lists: the support therapeutic management recommendations list. So there's one list that says yes, you should use this information. The second list is to indicate the potential impact of safety or response. And then there's this third list that's the pharmacogenetic associations for which the data demonstrate a potential impact on pharmacokinetic properties only. And so they're pretty much saying we don't really know if you should use this information. And of course, voriconazole and CYP2C19 are on this list at the bottom. But again, intermediate and poor, she didn't really know what this meant. And so she the team and she says, you know what, go ahead and start voriconazole at standard doses. And then they do, it takes about 5 days. And this is if you have in-house testing. If you have outside testing, you have to send it out. This could take a lot longer than just 5 days. So we got a patient with invasive fungal infection. We've now started them on a drug that may not be appropriate, but this pharmacist and the team didn't understand how to use that genetic test result. So 5 days later, it comes back undetectable. So you do what any good pharmacist would do, and you increase the dose by 20%. Then they wait another 5 days, and guess what? Still undetectable. So now you're giving this patient a fairly big dose. You're 10 days into therapy. You're still not getting therapeutic ranges of that particular drug, so you just switch drugs. But we've done 10 days now on inappropriate therapy on this patient with an invasive fungal infection. And so what really went wrong here? Why was this information not already in the label? So one thing, especially when voriconazole came to the market, PGX labeling was not mandatory. So these companies did not have to even study pharmacogenetics at this point to put it in the label. Post-market relabeling has a very high threshold for revisions. I think many patients actually have to die before they actually change a label. At least that's what it feels like. And then FDA labels are not necessarily up to date with new literature, right? Some of these labels were created 40 plus years ago, and they haven't really re-reviewed this information. So I've seen a lot of papers. I've actually reviewed some of these papers where they compare CPIC to these FDA lists. And I'm like, but you're really comparing apples and oranges, right? Our guidelines are up to date. We base this on updated literature. This is the better source. You get some pushback from that. So I always have to say, look, there's so many factors that influence variability in drug effects, right? As a clinician, you have to think about all these things, right? I mean, about, lactation. I don't have to worry about lactation knowing a 4-year-old, but an adult woman who's pregnant or just had an infant, yes, I absolutely do need to worry about that. So you really still have to individualize whatever you're doing to your patient, whatever you're prescribing to that patient. And let me tell you, there's no information in that FDA label in most cases about lactation and pregnancy and a lot of these other things, definitely renal function and liver function, but a lot of these, not much information is in that label. And this is the other point I always make. Again, I would never go pull an FDA label, right, as a clinician. I would go to Micromedics, UpToDate, PubMed, LexiComp, where they do have more updated information. Even LexiComp, even though a lot of what's on the label is written in there, they do have additional information, including pharmacogenomics for some of their drugs. But of course today, I'm going to talk about CPIC, but I will say this is how CPIC came along, right? We had this happening. We were having patients coming into clinic that had these test results, and we didn't really know what to do with them. But we knew the evidence was there. We knew it. So we just really needed to take it that next step. And when I say we, I really mean Mary Relling and Terry Klein, who started CPIC back in 2009. So I just want to make sure I give them credit because I thought it was just a brilliant idea to try to push this field forward. But I can't not acknowledge that there are other pharmacogenomic resources out there besides CPIC. I've already mentioned Pharm.D. They don't write guidelines, but it's a great place to go see if there is a guideline. So you can put that drug name in, and then you can look to see if there's any guidelines for that drug. But there's also the Dutch Pharmacogenetic Working Group, and there's also the Canadian group as well that also has some guidelines. And there's some other smaller groups that I believe have done a couple of guidelines here or there. And we are talking about maybe combining some guidelines with the Dutch Working Group here in the future, but so far we have not gone that route. But I am going to focus on CPIC because this is what I know. So CPIC was started in 2009. It was a shared project between Pharm.GKB and the PGRN, or the Pharmacogenomic Research Network. I started with CPIC in 2012, and we had about 60 members. And so since that time, we have grown to over 550 members from over 260 institutions, our members in 36 different countries. We now have 26 guidelines, but this number does not count all the updates we've done. In all, we've got 45 written guidelines, and some have just been updated online. And this covers now 25 genes, and with the recent G6PD guideline, we're over 100 drugs now, many of which are in the top 200 most used drugs. So one thing you have to remember about CPIC, I think there's two questions when you're thinking about pharmacogenomic testing. Is one, should I order the PGX test? And the other really is, should I use this PGX test? And how do I use this result? And that is what CPIC answers. We do not discuss the merit of actually doing the test, partly because to do that you really need randomized controlled clinical trials, and those are just pretty non-existent for pharmacogenomics. And there's good reasons for that, right? I mean, these are very rare events, very rare variants that we see. You have to enroll a whole lot of patients to be able to do this study. Nobody wants to fund them. That's the academia problem. And then, of course, would it be really ethical to have a group that you know you can test them and you're going to give them this drug that you know has the potential, and yet you put them in this control group? So it becomes a little unethical, especially for some of those gene drug pairs in which we do have a lot of. So this is why CPIC does it the way they do. I get asked all the time, are you ever going to go that other route? And I was like, show me the data. And it just doesn't exist. And for some gene drug pairs, I know there's a couple where we could make that argument. So again, these are based on the assumption that the test results are in hand and not to discuss the merits of doing the test. We do use standardized formats. So each CPIC guideline will look the same. They have the same sections. This has been the same since we first started. We do have a different system for grading the evidence and another system for grading the recommendations, and I'll go through an example of that. These are peer-reviewed. So if you are a CPIC member, and we do have many industry members that are CPIC members, so you're also invited, you do get a chance to review the guideline before we submit it for publication. We take that feedback back to our author group, and they can include it or cannot include it, depending, because they still get to make that final decision. because it's very important to us that these are actually, you can logistically use the guideline. And sometimes our members are like, you can't do this in this setting, and this is why, which is very good feedback to get before we go in for publication. And then, of course, it is submitted for in the publication review process. These are freely available. We are NIH funded, at least for now. And you can find them online at cpicpgx.org, pharmgkb.org, and also through PubMed Central. These are updated. I do try to stay on top of it. I get a weekly email from PubMed that I've set up these searches, and I go through each one of these. And I have staff that do some of it as well. But what we do is we go through it, make sure there's no new evidence that would change the recommendation in any of our guidelines. And this is done on a weekly basis. If there is a paper, I send it back to the authors, and they can immediately say no, yes or no, or this supports our recommendation versus doesn't support it. So it's a really great way to keep the authors involved, but also make sure we're staying on top of the evidence. We are cited in the genetic testing registry. These are filtered as practice guidelines in PubMed. So you can go to PubMed, put in clinical practice guideline, pharmacogenomics, and you should get a good list of the CPIC guidelines. We have a very strict authorship conflict of interest policy. And this is mostly because we follow, so the IOM put out standards for writing clinical trustworthy guidelines a few years back, and we follow those to the T, including their conflict of interest. So the steering committee for CPIC, this is what they help us do. We get all the conflicts of interest, and the steering committee makes that final call, but we do have fairly strict rules on who can be our guidelines. So as I said, we've got 26 guidelines covering 25 genes and over 100 drugs to date. I know this is a busy slide, and I would never let my trainees get away with this slide, but I think it's a good slide just to kind of show, and so you can see, you know, all the different genes that we have and all the drugs that it does cover. And then, of course, we just added G6PD. We updated the respirocase guideline, and now there's many drugs. And I will note that several of the drugs that we're recommending as a low-risk drug for G6P deficiency is actually a caution or a contraindication in the FDA label. So this might be the first time that we may have had differences in opinions, especially for these drugs. And I've boxed here on the right-hand side because I think as the evidence continues to grow and testing becomes more available, I try to stay on top of what genes are being tested. And so one of the things that we included in our opioid guideline update and also our statin guideline were additional genes besides just the typical ones that you see like CYP2D6 for opioids. We also reviewed the evidence for OPRM1 and COMT. OPRM1 had really strong data for a drug phenotype association. I mean, maybe moderate to strong. But the problem was it wasn't very applicable to practice, right? So I think the dose would have been an increase of 10% of morphine. I mean, 10% really isn't that clinically relevant, number one. And number two, no physician's going to start morphine higher because there's so much variability in the amount of morphine that a patient might need that starting the dose 10%, it just doesn't make sense. So yes, really great data to support a drug phenotype there with OPRM1, but really is just not clinical actionable at this time. COMT, there really was weak evidence for that one. And then for the SLCL1B1 guideline, we added ABCG2 and CYP2C9 for a couple of those drugs. We expanded that guideline, so it includes all statins now. And actually, I'm very proud of that guideline because they even went to a step further and took it to the guidelines the ACC guidelines, where they have the high risk, low risk, moderate risk, and they were able to really use our recommendation within that frame set. So I think it was just a really great guideline that those authors did a great job on. And then we also looked at HMGCR for statins, but it got a CPIC level C. There wasn't a whole lot there either. Okay, so as I said, you can get our guidelines at cpicpgx.org. And here's our guideline. There's this little tab at the top. I'm sure this has got a pointer on it. Little tab at the top up here that says guidelines. If you click on the tab, it will take you to a page that lists all of our guidelines. Each guideline has its own guideline. So you click on that guideline page, and here we go, voriconazole and CYP2C19. So this would have been really easy for that pharmacist to find. But knowing how to use the guideline may be a little tricky. But this page has everything you need. We've got the guideline in here, we've got the supplement, and then down here at the bottom, and I'll show you guys examples of these tables that are very helpful, I think, for many that use our guidelines, are what we call our implementation resources that have our little definition table, our little functionality tables, our frequency tables, and our diplotype to phenotype tables. So the process from the CPIC, from the very start all the way to the end, sometimes takes about a year and a half, and then our SSRI guideline that, oh God, I think we'll be submitting that one soon. I think that's taken about 2 years. So it just depends on the amount of evidence we have to review. There's sometimes conflicts with the authors. Sometimes the authors don't agree. So to come to consensus with the authors can sometimes take some time. We've argued over one word for an entire hour call before. So just that's how, I mean, I think it speaks to how much our authors care about these guidelines and how important it is to them to get it right. So even though I might have had a huge bruise on the top of my head from banging my head against the desk on that call, we still got through it. And I think even that whole hour of discussion, I think was still worth it. But it really starts with the guideline nomination. People always ask me, how do you decide on what guidelines? Okay, so we are really limited in resources. We can only move as fast as we can, and we do try. But because of that, we have to think really hard about what guideline we want to do next. And so we really take information. We take surveys from our members. They discuss them on CPIC calls. If somebody brings me a gene drug pair, I ask them to present it on a CPIC call to all our members, and they can decide and let the members kind of decide if this is one we want to move forward with or not. But once we decide to move forward with one, we have a selection process for our authors. So the authors are identified, and then again, we go through that strict conflict of interest policy, and then the steering committee makes the final call on the authors. And then the guideline is started. So I'm going to go into a lot more detail on this box here in a minute. But this is evidence review, which takes 2 to 3, 4 months sometimes, again, depending on how much evidence we're reviewing. We have many conference calls to talk through the recommendation table, and then the guideline is written around those recommendations. But once we have that draft, it's sent out to CPIC members, and then, of course, submitted. And we also post it on our website, and then it's submitted for publication. And then the final guideline is posted to the CPIC website, as I said. And again, we continuously are revising if needed. So I'm going to go into a bit more detail about what is in a CPIC guideline and how we get to that. So here you see this is a very simple, but I think this is really all you need to know about a CPIC guideline. If you understand this, then you can use a CPIC guideline. So we really have all the resources you need to translate that genotype to a phenotype to that clinical recommendation. So the important table here, one of the first things that we do is that genotype to phenotype translation. And this is a huge project for us. This is something that it does take a lot of our time and resources because I think this is very important. And it's important because our recommendations are based off that phenotype. So if we get that genotype to phenotype translation wrong, then our recommendation will also be wrong. So for our example before, we had that CYP2C19 star 17 star 17. This individual would have been an ultra-rapid metabolizer, and look, we recommended to avoid voriconazole. So this patient never should have received this drug. There are clear alternatives to voriconazole that should have been used in that patient. But it is with the moderate recommendation because it is based a lot on PK evidence. So let's talk a little bit about that genotype to phenotype. So it kind of really goes, there's a huge a lot of things we have to think about when we're putting together these tables and a lot of resources that we can use. So the allele definition table, that's that table that defines those alleles. So it tells you what a star 3 is, right? Because a star 3 for one gene is different than a star 3 for another gene, but it lays out the haplotypes for each one of those genes. I am so thankful for Pharm.D because this used to fall on us, but now Pharm.D does this, and we utilize those tables. And that, of course, is led by Andrea Gadick, and I think that she's done an incredible job pulling these groups together and helping us with nomenclature there. Another huge part, and this is on CPIC, is the allele functionality table. So the next step is we assign function to those alleles. Okay, this is very important because the function of the alleles, it's really the combination of the function that then defines that phenotype, and I'll show you that example in a minute. And of course, everybody's got 2 copies, so you have the CYP2C19 star 17 star 17, so we know that their diplotype is. Then you translate that to the phenotype, and then it really is that phenotype that we write the recommendation around. We also provide CPIC interpretation consult notes. So what this is you can use it in a lab result, you can use it in your EHR system to describe it. It's just an example of how you would communicate or give that consult about that test. So for our example, a CYP2C19 you need altered dosing recommendations for voriconazole, so just be that interpretation. And then we also provide, because we do feel very strongly that because this data really could follow a patient for their lifetime, I mean, with some exceptions in there. And so it's important that we have a way to make it so that physician goes in 10 years after the test is ordered, there's a way they can get that result right there at the site. So we also do provide some wording for post-test alerts as well. So here's an example of one of our table 1s. This is actually from the CYP2C19 guideline. And you can see that in our example here, it really is that function that was assigned that then designates that phenotype. So you have 2 increased function alleles, is that star 17. Another example might be the intermediate, where you have 1 normal function allele and 1 no function allele. I'm not going to get into those likely because we're hopefully going to get rid of those soon. But those are there now because we don't have great data on what to do with those decreased function alleles, and we needed a way to communicate that to our users. Okay, so we know how important the function is. This is what the tables look like. Again, this comes this is really provided, the information is provided from PharmVar. We put it in this format and it's posted online for the little definition table. And then this is our little functionality table. Some things that you're going to see changing over time is that we are adding a strength of evidence, and we're also adding a summary of findings. This feedback did come from the, we did submit for possible recognition of these tables to the FDA, and so we are working with them on this still, but this is one thing that we used to have this as optional, but now it is required for each one of these tables, so you're going to start seeing the summary of findings, and then you're also going to see the strength of evidence for each one of these as well. And then our diplotype to phenotype tables. I think CYP2D6 has over 10,000 possible diplotype combinations that you can have, so we do provide a full list of all the diplotypes then translated into that phenotype. So kind of back to this diagram here, talking a little bit about the recommendation. This is generally the table 2 in the guideline, but we sometimes add additional stuff because we need them, and it just, the numbers. But that table, so after we've done evidence review, so the evidence review is that first part, right? So we do the gene-drug association. So we pretty much search PubMed for any, we put the gene name in and the drug name in, and boy, we go through all those papers, and sometimes it can be in the thousands. That first walkthrough, and then we take the ones that do show or attempted to show a drug phenotype based on that particular variant, and we have a lot of observational type studies. We use preclinical, clinical studies because some of these variants can be so rare. We do have case reports, but I will say we won't write a guideline around only case reports, but we do kind of utilize that to think through the data. And then, of course, we have the in vivo PK and functional studies that we use as well. So sometimes that evidence can be high level, but when we start thinking about what I call actionability, you have to really think, how important is this test to have? Like, would you even use it if you had it? So we think about therapeutic index. So if you've got a drug, it's got a very large therapeutic index, which is on one side, you've got efficacy, the other side, you have toxicity. And really, these variants aren't going to cause much. They may be on the higher end for toxicity, but they're not really having bad toxicity to that drug. So really, the genotype may not matter. So we really look at drugs that have that narrow therapeutic index, because those are the ones where pharmacogenomics can probably have the biggest impact. And then the severity of the disease. How important is it that you get the right drug to the right patient at the right time? That's really important as well. And then we think of consequences of suboptimal prescribing as well. That kind of goes in with the severity of the disease a little bit. Availability of genetic tests, good genetic tests. And so we do have people on our guidelines that can kind of come back to us and let us know, this really isn't being tested clinically, and if they do, they're just testing this one variant. So that might be another reason we may not want to move forward with the guideline or recommendation. It's just not being tested clinically. And this is a big one, the availability of an evidence for alternative therapy. If we cannot recommend something different, or we would not recommend something different, then it's going to be a no recommendation. Because if you can't change therapy based on that pharmacogenetic test, then you can't use it. It's just, you can't use it. And we have run into this a couple of times, and you'll see this, I think, on the upcoming SSRI guideline as well. You just can't cut out an entire class of antidepressants. You just can't. So when you get that guideline, please. So let's go through an example of how we might think through this on a CPIC guideline. So we know, and there's great data to support, that voriconazole is extensively metabolized with less than 2% of the original dose excreted in an unchanged form. And the main circulating metabolite, the voriconazole N oxide, has 0 antifungal therapy. So think about our patient who was ultra-rapid metabolizer, who just really very quickly metabolized the voriconazole, so they really didn't have any active antifungal agent on board. So let's go through our list of things that we thought about. So the therapeutic index for the drugs. Yes, there is it's not, I wouldn't call it too narrow, but there is a very defined therapeutic index for voriconazole, so at least that it really does help with that recommendation. We do see patients that have toxicity, sometimes at the higher ends and sometimes actually at the low ends, but we do see toxicities if they do step out this one area of the concentration. And then of course the ultra rapid, of course, on the other side, if they're metabolizing too quickly, it's not going to work. Then you have the severity of underlying disease. In this case, either you're preventing an invasive fungal infection or you have an invasive fungal infection. And then we have the consequences of suboptimal prescribing. Of course, you can die from an invasive fungal infection. This is what my Ph.D. was on, and I still can't say it. Severe or serious morbidity. And then availability of genetic tests, CYP2C19 is widely tested. So you may have ordered CYP2C19 for clopidogrel, but now you have it in the patients on voriconazole. So it's something else we have to think about. And then the availability of evidence and for alternative therapy, and absolutely there are many antifungals that have been shown to be just as effective. So definitely, if you can avoid the drug, you should. So I want to talk a little bit about the evidence and how we see this. So here's an example of some of the PK data. So over here you've got the voriconazole concentration. It's been normalized by dose, and down here you can see these are the diplotypes. And I kind of want to show you this because now we have pediatric recommendations, and this is a good example of why we have pediatric examples and why you may not be able to just take the adult recommendation and use it. So in adult, it's pretty clear, right? Patients that are ultra rapid or rapid metabolizers definitely have, definitely don't, they really have low levels of circulating voriconazole. And then you've got your intermediate metabolizers, not much of a difference there. But then your poor metabolizers, you definitely do see an increase in concentrations in adults. And again, the study was fairly small, but I think it still demonstrates a bit about what we see. And so in the evidence table here, we were able to use this information for adults, and we were able to say, yes, that definitely there's a moderate level of evidence that really supports that there's definitely a PK issue here. And you can see here, and I like this example here, because the way we do evidence review is we have 2 to 3 authors, depending on how much evidence we have to review. They independently review this. And then we have multiple phone calls to talk about what we call that consensus. So that one you see in the guideline. In our final, then you only see that last column here. But let me tell you, sometimes that to get to that consensus can take a bit of time. So if you see a high, that means the evidence includes consistent results from well-designed, well-conducted studies. So I always think if you were to repeat, if you were to do another experiment the same, would the direction or the effect of the magnitude change? And if you think not, we're going to give it a high. Moderate is where you might have evidence is sufficient to determine the effects, but the strength of the evidence is generally, and this is generally limited by number, quality, or consistency of the individual study. So again, I think maybe maybe we know that voriconazole, you're going to have less voriconazole levels, but maybe we don't really understand the magnitude of that. And so that would get a moderate, and weak is where we can't really make any decisions at all based on that evidence. And then we have the CPIC recommendation table. Again, this is where a lot of discussion, especially in the wording, goes into these tables. And so you can see here, this is what the voriconazole one is for adults. And you can see here for ultra rapid, again, you choose an alternative agent. For poor, we also said choose an alternative agent because we have to really think through if you actually had to send this out. So not all hospitals do voriconazole testing inside. So they send it out. It can take 2 weeks sometimes for that level to come back. And so to have your patient that's at too high of a level of voriconazole for that long a time can be dangerous. So we do have recommendations for both. If you can avoid it, just avoid it in those patients. But if you can't, you may want to start with a lower dose. But we don't know what that dose would be. That data was not available at the time of the guide. So now we're going to look at pediatrics. So you kind of do see a little bit of the same with ultra rapid. They do definitely have decreased or increased metabolism of voriconazole. But when you look at the rapid metabolizer, it looks very close to what the normal metabolizer, and this is not the only study that showed this, and that's because there's so much variability in pediatrics, right? And maybe in this case, pharmacogenetics may not matter that much in these patients. And so for that, and I think you can kind of see, it kind of goes along the same with the adults for the IM and the PM, but we did take this into consideration when writing the recommendation for pediatrics. We just said it's probably okay to use. You would just need to treat them like you do a normal metabolizer. And then so we have the strong, moderate, and optional, and the no recommendation. So strong is where we think the evidence. So remember that Venn diagram. We've got the evidence on one side and actionability. And so that strong takes into consideration the evidence level and also the actionability. So the strong recommendation, the evidence is high quality, and the desirable clearly outweigh the undesirable effects. Moderate, there's a close or uncertain balance as to whether the evidence is high quality, and the desirable clearly outweigh the undesirable. And optional is really either we're basing it off extrapolations or we kind of have weak evidence, but it still kind of makes sense. Like if you have an alternative drug that it costs the same, it's usually on formulary at hospitals, maybe it just makes sense since we don't know just to optional, just maybe avoid that drug if you can. And then the no recommendation. This has been used quite a bit lately, more so than it did when we first started CPIC, and this is where there's just really insufficient evidence or confidence or agreement to really provide any course of action at this time. Okay, so I'm going to switch gears just a little bit and talk a little bit about where I hope that our field can go and where we can grow, and how to get us there. Because I think the regulatory bodies, they like standards. They like standardization. And so when you've got one laboratory reporting the same diplotype, but then saying, you know, giving it a different phenotype than another laboratory, that is not standard. It's confusing. The terminology is a big part, y'all. Y'all, I just said y'all, I'm so sorry. I get too comfortable up here sometimes. But I think the more standard we become, the better our field can really forward. And so in 2018, and not to push my paper forward, but I thought this is exactly how I was thinking about this when I was putting these slides together. But there was this figure in this paper that we outlined really, okay, the current aim is to improve standardization in the processes required for implementation of PGX. What are those key drivers? And this might impact a lot of you in this room. The standardization of the clinical laboratory, and you can see that takes a lot over here in the right-hand column. These lines go to a lot of these listed here. I'm not going to go through them all for time, and then you have standardization of results reporting. So the reports really do need to look the same. We should be reporting the same result for the same, the same patient. The same patient should send the report out to two different labs and get the same answer back. That needs to happen. And then standardization of test ordering and reimbursement. And so we have made some strides, I think. Vicki Pratt sitting in here and her work with A&P with the minimal set of alleles integrated into the PGX panel. So it's just a list of alleles that you should be testing if you're going to test. That's important. That will help also with getting the same result from the patients because you're all testing the same variants. The development and maintenance of PGX nomenclature. So this is very important too. We really need to be calling things what they're supposed to be called. And so PharmVar is the great resource for that now. And then CPIC. So what we've tried to do is work the best we can. Our guidelines use standardized terminology where we can. We had a term standardization project that we did a few years back, and we utilize all those within our guidelines. We submitted those phenotype terms for SNOMED and LOINC, which means that people who are implementing and use SNOMED terms, they can pull those straight from there. They don't have to go creating their own fancy terms. And then the genotype to phenotype translation. So again, we provide that, and if labs would if you're going to use the CPIC guideline, because those CPIC guidelines are really written around how we translate those diplotypes to the phenotypes. So I've seen labs use the CPIC guidelines, but not then translate the diplotype to phenotypes the way CPIC is recommending. And so you're not technically using the CPIC guidelines. So just kind of keep that in mind. That translation is a very important part of what we do. And I also think the standardization here, so there's a big push on educating physicians and pharmacists. But honestly, all that stuff about CYP2C19, star 17, translating it to that phenotype could go away. They don't have to understand that. All they need, if we can get standard, if we can be standardized, then that physician only needs to know that patient's an ultra-rapid metabolizer and this is what you do, right? But we can't right now because we have to say, well, what variants did they test? You have to go through this list of things before you can utilize a report to know if it's accurate or not. But if we're more standard, then that education can go away and it can be super simple, just like creatinine clearance. You have a drug, the patient's creatinine clearance is less than 50%, you give them half the dose. We could get there with pharmacogenomics if we could be a bit more standard in how we're reporting. And then we have the pharmacogenomics working group that's coming up. We started some work on this already. This is being led by my co-PI, Terry Klein, also with Stuart Scott and Michelle Worrell-Carrillo. I think this is a great group of leaders. I think they're going to do some good here. But this is really with the mission. I think it's going to be really important for the future for us to really be combined maybe with the bigger group of ClinGen. So you're going to start seeing more PGX within that ClinGen database. But they have a framework of tiered standard terminology and definitions this is what we're going to be creating that reflect clinical significance. So it will be something like the pathogenic, not pathogenic. That doesn't work for pharmacogenomics because it's not a disease in most cases, right? And we think that's very misleading. So we're going to come up with new terminology that really shows clinical significance for drug response variability. And then we're going to help hopefully facilitate some of the incorporation of pharmacogenomics knowledge into the ClinGen database. So that work's coming. I'm on 3 working groups for that, and I'm very excited about that work. And we also have Stripe. I'm somewhat involved with Stripe, kind of paying attention to what's going on there, but this is the group that's able to talk to the FDA. They set up a collaborative community with the FDA. The point of that collaborative community is to be able to provide feedback to the FDA and actually have some open conversations with them about how we can really move forward. And you see standardizing laboratory practices. So they are working towards hopefully getting some standards put out that we can all follow and hopefully push it forward and be more standard in the way we're reporting out these results and interpreting these results. I went faster than I thought I would, but that's okay. But of course, it takes a village. I wish I could explain to people the hours and hours and hours of time that our authors do dedicate to these guidelines. I mean, when you've been working on the same project for 2 years, a lot of work goes into that, and that's what our authors sometimes do. Andrea Gaedick's been huge, and of course helping us with those nomenclatures and really with the little function stuff, and then of course the whole team of investigators at CPIC. I'm just here representing that team, because it really is great to work with all of them. So I think that's it for me.